Hello and welcome back to the Comparative and Omics Tools in BioCyc webinar series. This is Part 3, Comparative Genomics. I'm Alexander Shearer, speaking on behalf of the Bioinformatics Research Group at SRI International, and I'm showing off some of the features and tools available on biocyc.org. In Part 1, we introduced you to the cellular overview and showed you what you could see there. In Part 2, we showed you the cellular omics viewer that lets you look at omics data on a cellular metabolic overview. In Part 3, we're going to look at comparative genomics tools in BioCyc, specifically ways you can compare across different organisms or ways you can get some interesting information out of a single organism. At the end of Part 2, I told you we would be moving on to comparative analysis, and I pointed you to the link down here on the query page, so we'll click on that now. This takes us to the comparative analysis summary table page. This page lets you generate summary tables for a single organism or for a number of organisms, comparing one or more traits or sets of characteristics across those organisms. So, let's take a look at the kind of comparative analyses we can do. We can look at reactions, pathways, compounds, proteins, transporters, and transcription units. And we're able to do this across one or more of the organisms from the entire BioCyc collection. So that's our Tier 1, 2, and 3 organisms. Now, this is a wide range of possibilities. There's a lot you could do here. The sort of simplistic example that I like to use as a start is, say we're trying to design a drug that will target tularemia, so Francisella tularensis, but we don't want to hurt the person or their normal bacterial flora. And say we're interested in looking at reactions we might be targeting. Well, so we're going to click on reactions, so that's what we'll be comparing. And note, you could compare more than one thing at the same time, but I'm going to stick with reactions for now. Then we just need to go down and click on a couple organisms from this big selection. So we have Homo sapiens, so we have us. And scrolling down some more, we have E. coli, which is my simplistic stand-in for normal bacterial flora, and we have Francisella tularensis, our nominal pathogenic bad guy for this example. So having chosen our 3 organisms we're going to compare this time, I'll click Submit. And what we get is this kind of comparative analysis summary results page. And as the warning at the top here shows you, you're not only looking at differences in biology here, you're looking at differences in curation. And this is a good example because E. coli K12, this is BioCyc we're looking at. That's a Tier 1 heavily curated database. It's going to be more populated and probably more accurate than a lot of the automatically generated databases. And the Homo sapiens and Francisella databases have received some curation, but not nearly as much as E. coli. And of course, the entire Tier 3 collection has had only manual QA and no manual curation. And as it tells us here, any cell of a table that is a hyperlink can be clicked to see more detail about the contents of the cell. I'll show you kind of what that means. And so what we get here, we get a number of breakdowns for a reaction analysis. We get a breakdown of reactions by type. So this is a high-level view of each organism. So we have Homo sapiens reactions in which all substrates are small molecules, about 1,000 in E. coli, and about 600 in tularensis. And again, this probably reflects curation differences. Transport reactions definitely reflects curation differences, and so on and so forth. Reactions of small molecule metabolism, and you see total numbers, numbers with identified enzymes, numbers without total EC numbers because you have partial EC numbers, and so on. And then a breakdown of reactions by top-level EC categories, and then finally a distribution of isozymes across the different small molecule reactions, which is to say, what percentage of your reactions have, well, let's look here, a single enzyme catalyzing them, 2, 3, more. We can see that Homo sapiens, as represented in our HumanCyc database here in the BioCyc collection, has 20 reactions for which there are 10 or more isozymes for each reaction, which is pretty impressive. E. coli tops out with 2 reactions that have 6 to 10 isozymes, and Francisella, as we have it, tops out with 1 reaction with 5 isozymes. So that's all kind of useful high-level information, but you might want to take a closer look. And you could, for example, click on one of the categories. Say we're interested in looking at oxidoreductases. Clicking on that link generates this table of all the oxidoreductase activities across all 3 organisms. So it's the union of the available oxidoreductase activities. And what we then get is a link, and each one of these links will actually take you to a detailed comparison across all organisms. I'll do that in a moment. And over here an X indicating whether the thing is present in all of them or not. We can see our top set of oxidoreductases, they all have it. And if I click down, we can see that maybe oxidoreductases are not the best drug target in our fictional drug development that we're imagining. With that in mind, let's return to the original page and try a different category. Back to the original comparison page. My organisms are still selected. Let's go back to the top of the page. Instead of looking at reactions, let's look at functional units. Let's look at pathways. So I've clicked pathways. I'm going to page down again. And once more, I'm going to submit. And now we're comparing pathways across coli, Francisella, and Homo sapiens. So once again, this takes us to a comparative analysis summary results page, this time for pathways. And so we get a breakdown by pathway class here. So you can see we have different categories. So say we pick a category like fatty acids and lipids, and we're going to click on this. And it takes us over here to a comparison by pathway. And now we can see a couple of different cases. For example, we can see a case for a pathway that's present in all 3 organisms, elongation of saturated fatty acids. So let's click on that. And this is a pathway comparison. Now this is important because this is also a comparison that you can get to from each individual pathway. So actually, let's go to an individual organism's pathway page very quickly and see how you do that. This is the fatty acid elongation saturated pathway page for E. coli. Now say we wanted to do a cross-species comparison just looking at this pathway rather than comparing across the whole organism. Well, each pathway page has this cross-species comparison button, so you can click on that and it will take you to this kind of comparison. Now in this case, because we preloaded the species we were looking at from the comparative page, they're already selected. Otherwise, it would have taken us to a selection page.